your biology class according to class 12 syllabus of NCERT, and in the series of classes, in the last class we have discussed about agrogel electrophoresis. Today's class we will going to discuss about the PCR, okay, because the result of agrogel electrophoresis is that we got the fragment, right, or our desired gene after isolation or purification. We got the fragment of the desired gene, and now what? That we need to make multiple copy of that. Why that is necessary? Because what in the last class we have seen is that the process is very tedious, costly, time taking, because you need the organisms, you need to degrade that organism, you need to isolate from the genetic material. So it is a quite tedious and yes, a time consuming process. So we should find such a way that once we can write notes. Suppose in your class teacher provide you have any handwritten notes. What you will do? That we make multiple copy of that. Yes, suppose tomorrow is your exam and you got a notes. So I think it's not time that you are going to be, everyone going to take that original notes and start writing time one by one. Yes, not possible. What we do? We make a xerox. Yes, and easily everyone get. We can say that PCR also do the same with the isolated desired gene that we have fragmented, okay, by the agrogel, we have extracted by the agrogel electrophoresis. So that is the reason why PCR is necessary. So we are going through with the topic, okay, and yes, focus here. What is PCR? PCR is actually a type of reaction or process, okay. It is a process which is known as polymerase chain reaction, okay. I have written this in short form. Polymerase chain reaction, okay, where we perform these things in a machine that is known as thermocycler. It is Gary Mullis who given this technique. Okay, so PCR is a process known as polymerase chain reaction, which takes place in a machine known as thermocycler. Now I told you what is the use. It is used to make multiple copies of desired gene. Okay, the gene that we have extracted, okay, or isolated, that can be multiplied or amplified, okay, numbers of copies by using this method. So this is what. Now what are the steps actually there in the PCR? Okay, what are the reactions actually takes place in the PCR inside the thermocycler machine? Okay, so let's focus on that. See, the sample that we need to be copied or amplified is actually a double stranded structure. Yes, double stranded structure, and we all know that in the replication, yes, we have completed with those topics. In replication, what happens? The double-stranded DNA gets separated into the single-stranded. Yes, occasional fragment, leading ligand strand, these things are going to be formed. Yes, so hydrogen bonding which is present between both the strands is going to be ruptured, and they are going to become single-stranded simultaneously or slowly. It's not like all the DNA gets separated and then the process. Actually, it is one side it is getting separated, and another side the process of replication going to be takes place, and finally, when the genetic material completely separate, you will finally simultaneously you will get the double stranded cell structure. Also, because it is side by side process. In one side it is going to be opening and another way continue in the forming the double helical structure. So what are those process or steps involved in the PCR? First is your denaturation. I will come to this point. Denaturation. Okay, denaturation. Second is your annealing. Third one is your first one is denaturation, second one is annealing, third one is extension. And the result of all this is actually finally amplification. Yes, finally it is your amplification. The first step denaturation, then annealing, then extension, and then amplification. What happens in denaturation? In case of denaturation, okay. So in the case of denaturation, what happening? This double stranded structure, which is actually 5' to 3' and 3' to 5'. Correct? This is the what our genetic material have. Denaturation means what will happen? This double bond, this hydrogen bonding, okay? Hydrogen bonding which is there in the both the strand is going to be break down slowly, and what we will get is like this. In this way, two strand, denatured strand we will get. Okay? Now suppose this is the region of amplification. Okay? This is the region of amplification. This is need to be amplified. So what will be happen? This one is 5' and this one is your 3'. Okay? And this one is 3' and 5'. So we all know the direction is opposite 5' to 3' to 5' direction from hydroxyl group. So again what will be happen here? A small nucleotide sequence, oligonucleotide we known as primer. Okay? We known as primer. These structures are known as primer. Okay? These structures are known as primer. So annealing is actually joining of the primer to the 3' region. Okay? Annealing is the process of addition of the primer or oligonucleotide to the single stranded. Now what will happen? There is so denaturation done, okay, and after that addition of primer, that is your annealing. In extension, what happens? This structure start going to be continued. Okay, the process of replication we can say, or this new strand start going to be formed. This new strand start going to be formed. Now for this what they needed. So the process of extension, what needed is DNA polymerase, okay, plus deoxynucleotide, okay, deoxynucleotide, because what we are going to be add in the single strand is your nucleotide. Yes, that is we have used DNA, so deoxynucleotide. Now DNA polymerase, it is stack polymerase. The DNA polymerase that we have used is actually obtained from Taq polymerase. Taq stands for Thermus aquaticus. Okay, Thermus aquaticus. Okay, so we obtain the DNA polymerase for the in vitro condition from the Taq, that is Thermus aquaticus, and we add deoxynucleotide with that. As a result, what happened? The extension leaves are going to be joined in the primer, and the extension of the strand start taking place. Okay. So after this, we set cycle that how many cycles of this particular system is going to be take place, because suppose one cycle takes place, what will be happen? One double-stranded structure separate and form two. Again that cycle, if both are going to be separate again form, so it will be the percentage of equational division, right? So suppose if we made 30 cycles or something like that, in that we get near about approximately 1 billion numbers of the primer. So this is the amplification in the final stage, that we had how many cycles, okay, how many cycles of this reaction PCR, how many cycles of this polymerase chain reactions takes place in thermo cycle, and depending on that, after the completion, okay, we will get the amplified gene, okay, amplified fragment of what? We will get the amplified fragment of DNA, and that is going to be same like our desired gene or desired DNA that we have isolated. So in this way we can increase our desired gene numbers, okay, without going to the process of electrophoresis and DNA isolation again and again. So in this way, this is a very much beneficial for the biotechnology, okay, because we can make multiple copies and we can store for our future research and analysis. Okay, so hope you people understood the concept of the what polymerase chain reaction. Okay, now the next that we need to study is how we are going to insert this in our code. Please give me 1 second. Sorry for the interruption. Just 1 second. Now, so what happened now? We got our particular copies of that particular desired gene. Now what we do? We got the gene. Now we will insert desired gene into vector. Correct? We insert our desired gene into vector, and this is nothing but the plasmid. Okay? This is nothing but the plasmid. Yes? How this is going to be occur? We treat our desired gene with a specific restriction enzyme. Correct? Specific restriction enzyme is used for both desired gene plus vector. Both are going to be treated with the same restriction enzyme, because we need common means insertion site, because we know that the restriction enzymes are specific, they are cut site here. So if we use same kind of restriction enzyme in both the desired gene and in case of vector, so we will get the sticky end, and after that with this treated fragment, that is by using restriction enzyme, treated fragment plus DNA ligase. Correct? We use that treated fragment with the restriction enzyme plus DNA ligase, and we transfer that into the plasmid. Yes, we insert that sequence into the plasmid, and what will be happen with the help of DNA ligase, that particular fragment, okay, that particular fragment of gene gets inserted into the plasmid. Okay, get inserted into the plasmid. Got it? We will get this recombinant DNA. In this way we got our rDNA. Okay, in this way we will get our rDNA. Now what is next thing? Okay, the next thing is that insertion of this DNA, recombinant DNA into the host. Okay. Now, one thing people should remember, I think yes, we have discussed also that plasmids are resistant to specific or different types of antibiotics. Yes, resistance to antibiotics. All plasmids are not resistant to every kind, but yes, they are specific to the types of antibiotic. Now, our DNA is ready, recombinant DNA. Now we need to be transfer this into the host. So, what next that we need to do? Now the second thing, is there any gate in the organism or host organism that it get open and you put the plasmid inside the host body? Yes, is it so? So what is the method? How can we insert our recombinant DNA or the constructed plasmid into the host? So there are some methods. There are some methods that we use in the case of transfer of the desired plasmid now or recombinant DNA into the host. We make the cell competent. Sorry. We make the host competent. We make the host competent so that it uptake the foreign gene. Yes, because the gene that we are transferring or editing is foreign, so we make the host competent. Okay, so that it uptake the foreign gene. How this is possible? We generally do heats of treatment. Heats of treatment. Okay, in this what actually happens, we keep the host cell for some time in the cold, very cold temperature, and suddenly we will change the temperature to the high temperature. That causes development or formation of pores in the cell wall or in the membrane of that particular host. Okay, so that the plasmid can be get entered, or we can use the enzyme polymerase. Okay. We can use the enzyme polymerase which increase the pore size in the membrane, cell membrane, and allow the plasmid or the foreign genetic material to get entered inside the host. Now what will happen? The host is provided a suitable environment for the division, for the cell division growth. Okay, and later on we will check for the success of this competent, that transformation. The transformant that we have transferred the desired gene into the host, that is successful or not. For that, the step that we perform is screening test. Okay, that is known as screening sort. So in the next class we will discuss about the screening methods, that how we are going to check whether the recombinant gene that we have transferred in the host is successful or not, whether they have forming our desired product or not. Okay, and then yes, the antibiotic resistance property that we have discussed in the case of plasmid is going to play very much important role. So hope you people have liked the class and got the concept. And yes, if you people have any doubt, okay, if you people have any doubt regarding the topic, leave a comment in the comment section. And yeah, further we will again meet in the next class. Okay, so thank you and have a nice day.